Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the actin filament. Okay, so before going to this topic, I want to say that you just check out my previous videos related different topics about biological science, and obviously I'm thankful to all the subscribers that they help me for the growing of this channel. Okay, so actin filament is what? This is made up of G actins. This globular G actin is 43 kilodalton or kDa ATPase, and like tubulin, it has a binding site for a nucleotide, but only ATP or ADP can bind with that, not GTP or GDP. And why ATP or ADP is needed, we will just learn in the next para, because the hydrolysis of the ATP or the formation of ADP is needed for the polymerization process. Okay, so here just we will learn here that actin filament is made up of G actin, and it has a binding site for the nucleotide. Obviously, it has a magnesium ion and complexed with either ATP or ADP. That means if this magnesium or bivalent ion is not present, then the ATP and ADP cannot bind properly with the G actin. And ATP, ADP and magnesium ion all bind with G actin, form a complex. The G actin subunits that assemble head to tail to generate filamentous F actin. When G actin subunits, that means small G actins, all bind with each other or assemble with each other from head to tail, then filamentous F actin is formed. But it has the structural polarity. It has structural polarity means one pole is minus and another pole is plus. And the two parallel F actins twist around each other in a right-handed helix, just like this. Okay, just like this. You can see. Okay. So two like this one is one F filamentous actin, and this is another F filamentous actin. Okay, they twist with each other and to form right-handed helix. Okay, that means one right-handed here you can see that the two helix are there, but that means two strands, two filamentous F actin strands, they just twist with one each other. Okay. And the structural polarity of actin filament is created by the regular parallel orientation of all their subunits. So here the subunits like that, these are very small and these are very small in that way. Okay. Okay. Just like this. Okay. So regular parallel orientation of the all their subunits, and the actin filaments are thinner and more flexible, usually much shorter than the microtubule. That means these are very flexible, shorter and thinner. And actin filament, this is a polar structure with two structurally different ends. A slow growing minus end and the faster growing plus end. Okay, slow growing minus end and the faster growing plus end. This is the minus end and this is the plus end. So what is saying in this paragraph? That G actin subunits, they assemble from head to tail and form the filamentous F actin, and the filamentous F actin, they want two strands, they twist with each other and they form the right-handed helix. These are very filamentous, filamentous F actin. These are very flexible, shorter and thinner, and it has the two pole. One is the minus pole, another is the plus pole. Minus pole, this is the shorter growing pole, and the plus pole, that this is the faster growing pole. And ATP hydrolysis here important for what? Actin polymerization. For the actin polymerization, ATP hydrolysis is needed. And another thing is that the minus end is called the pointed end and the plus end is called the barbed end. Next, that is the polymerization of G-actin. The polymerization of G-actin, this occurs in three phases. One is the nucleation, second is the elongation, and third one is the steady state phase. The nucleation, that is the G-actin aggregates into short oligomers. So nucleation process is what? The aggregation of G-actin is occur here. Okay. Elongation is what? Nuclei are rapidly elongated by the addition of G-actins. So here the nuclei, these are elongated by the addition of the G-actin at the both ends. Okay. And third one is the steady state phase, that means the equilibrium is reached between the filaments and the monomers. Monomers means subunit here. And as the F-actin filament grow, the concentration of the soluble G-actin monomer decreases. Okay, this is important. Note that when the F-actin that becomes grow, then the G-actin monomer decreases. Concentration of the G-actin monomer because G-actin monomers attach with this and form the F-actin filament. Okay, so that's why the concentration of the G-actin monomers in the solution that becomes decreases. Okay, becomes decreased. Next one, the two classes protein very important for the nucleation, and it is called the spontaneous nucleation. Why the spontaneous nucleation? Because here trimeric nucleus formed. Trimeric nucleus formed, and nucleate actin assembly. Here, the nucleate actin assembly and the nucleation, both cases, the proteins are very important role play. They can play important role both cases. One is the nucleation and another is the actin assimilation. Okay. Next, these are the actin nucleating protein. And name of this protein what? One is the formins and another is the actin related protein or the ARP 2/3. Okay, the two proteins are very important. One is the actin related protein, that is the short form is that ARP 2/3, and another is the formins. And another 5 proteins also important: P40, P35, P19, P18 and P14. And the complex of ARP23 formed with the ARP23 and 5 other proteins. And now we will know why this complex is needed, or the formation of this complex is needed, because this complex bind to the side of the existing filament and initiate the polymerization of new filament. Clear? If this complex is not bind with the sides of the existing filament, then polymerization of the new filament is not possible. Okay, and this polymerization of the new filament with the existing filament, that occurs at 70-degree angle formation. Clear? And another two important, FH1 domain and FH2 domain, these are the part of the forming. First, FH1 domain that interact with the prolifin and providing G-actin for filament assembly. So filament assembly, for filament, which filament? G-actin filament assembly, the prolifin is important, and this bind with the FH1 domain of the forming. Without this binding, the assembly of the filament is not possible. Clear? And for this is regulated. Okay. And FH2, that nucleates F-actin. FH2 is another domain, obviously the part of the forming. It helps in the nucleation of F-actin. Clear? And forming is regulated by the Rho GTP. And total function of the forming, that is regulated by the Rho GTP. So this is the actual actin filament, and what is the role of these and how the muscle contraction is occurred. So these are another important topic, and we will learn in another video. But again I have to explain about the myosin. What is the important role of the myosin is very important for the muscle contraction. So here we will learn about here the actin filament, and in the next topic we will learn about the myosin, and then we will try to learn about the muscle contraction. Okay, so please continue with my channel and support me a lot. Thank you.